Breakthrough Alzheimer's Map reveals hidden genetic drivers. A groundbreaking study has produced the most detailed genetic map of Alzheimer's disease to date, revealing critical connections between genes that likely cause the disease's progression in the brain. This isn't just another correlation study. Researchers have identified potential cause-and-effect relationships, opening new avenues for targeted treatments. Uncovering the molecular cascade for years, scientists have observed which genes change in Alzheimer's patients, but not how those changes drive the disease. This new research from UC Irvine and Purdue University changes that. By analyzing brain tissue from over 272 deceased individuals with Alzheimer's, the team used a machine learning system called Signet to map out gene activity in 6 key brain cell types: excitatory neurons, inhibitory neurons, astrocytes, microglia, oligodendrocytes, and their progenitor cells. Signet doesn't just show which genes move together, it identifies which ones actively influence others, the hub genes that control entire networks. The tool's ability to analyze both individual cell behavior and broader genetic patterns is a major step forward. Excitatory neurons. Ground zero for disruption, the study revealed that excitatory neurons, critical for brain signaling and memory, exhibit the most severe genetic disruption in Alzheimer's. Researchers identified nearly 6,000 cause-and-effect interactions within these cells, suggesting that these neurons are the primary battleground where the disease takes hold. Different types of brain cells play distinct roles in Alzheimer's disease, but how they interact at the molecular level has remained unclear. Our work provides cell-type-specific maps of gene regulation in the Alzheimer's brain, shifting the field from observing correlations to uncovering the causal mechanisms that actively drive disease progression. Min Zhang, UC Irvine epidemiologist, validating the findings to confirm the results. The team tested their map against additional brain samples. The same genetic chain reactions were observed, strengthening the evidence that these disruptions aren't random. This level of validation is crucial because complex diseases like Alzheimer's often involve multiple contributing factors. Clear, reliable data is essential to identify true drivers. What this means for future treatments, the identification of both critical hub genes and the widespread disruption in excitatory neurons presents specific targets for future drug development. While treatments are still years away, this map provides the most precise roadmap, yet for interventions aimed at stopping or reversing Alzheimer's progression. However, the study doesn't definitively prove these gene changes cause Alzheimer's. The next step is comparing these patterns to healthy brain tissue to isolate the disease-specific shifts. Nevertheless, this research is a critical step forward in understanding a disease that affects millions worldwide. The Alzheimer's field has long struggled with complexity. This study offers a level of clarity that could finally unlock meaningful progress.